So here is our last topic on genetic variability. It can occur by mutations. So we'll take a look at this in a second. Again, remember that as you listen to this video and watch what happens on screen, you will be prompted to ask or answer different questions on the video, and you're also writing those questions in your notebook and answering them. Then at the end, you'll record the chart in your notebook on the specified page that will be given to you by the teacher or on the board. So let's remember what we mean by genetic variability. By genetic variability, we mean that the genetics, the traits that organisms have, are all different, and those can happen because of something like crossing over, where we exchange segments of DNA called genes. Those genes can have different alleles. So, for example, the gene for eye color can have an allele for brown eye color or an allele for blue eye color. And now, lastly, mutations can occur. So, some examples of genetic variability here. In our class activity, we learned that somebody who has 6 fingers, it actually has that dominant phenotype. And so if they have 6 fingers, they will pass 6 fingers on to their children. However, we don't often see very many people with 6 fingers because 6 fingers can kind of get bred out. If the majority of the population of people have 5 fingers, then they're going to continue to pass on the trait for 5 fingers, meaning that nobody else will have 6 fingers unless somebody with 6 fingers starts breeding into the population. So, when we think about mutations, a lot of us think about people like mutants. So, somebody like people in X-Men. So, for example, we got Wolverine up front here. Wolverine was born with a mutation where he had bone claws, but he also didn't really age and he healed really fast. So he obviously aged into this man, but he was able to heal really, really, really fast. Or an example like Spider-Man here, where he was bitten by this random spider in a lab that I guess was radioactive, and all of a sudden he started to have these mutant superpowers like the radioactive spider. So he's clinging to the wall there, shooting a spider web. These things obviously don't happen. Let me give you some examples of real mutations that can occur. So here we have this young boy who has a mutation called progeria. In progeria, it is a mutation that affects the nucleus of his cells, which causes him to age faster. And people who have progeria have a lot of other health problems as well. They have problems with their bone structure, and obviously they age faster, and they typically don't live past their teenage years, maybe sometimes into their 20s. And so obviously a mutation like this is not very beneficial. It's not very helpful to them. Down here we have this little kid that's holding up these weights that obviously a little kid of 3 or 4 that he is probably couldn't usually hold up. He has, I guess it could be considered more a beneficial mutation, where he builds a lot of muscle. And that's because he has a mutation in his body where he is not able to produce a protein that inhibits or stops muscle production. And so because his body cannot stop making muscle, he just keeps making more and more muscle. And so one drawback to this is that he has to consume a lot more food to keep up with the muscle production that his body is wanting to do. So let's begin our chart here. By mutation, we mean that there are changes to the DNA nucleotides. So remember, the nucleotides are those letters either A, C, G, or T. And so maybe a normal DNA sequence looks like this, A, C, G, T, but several different mutations can occur. One mutation is where a nucleotide gets inserted. It just gets put into this other sequence. So notice here we have insertion in green, and this T got inserted into our normal nucleotide sequence. Sometimes a nucleotide can be deleted, so a deletion mutation. So notice here we're missing the nucleotide G. And then lastly, there could be a substitution mutation, meaning one of the letters gets substituted or changed. And so here we have the C changed to a G. Some causes of mutations. Mutations happen all the time. They happen naturally. And so as your DNA gets doubled, it gets copied during interphase. So remember our cell cycle, we learned this during mitosis, and during the cell cycle there's this little teeny tiny area where mitosis actually occurs, but the rest of the time it's in interphase where it's preparing to go through mitosis. And part of that interphase is the S phase, meaning this is where DNA gets doubled. And so during that time, as DNA is replicated, it happens very, very, very quickly, which means mistakes are bound to happen. So there are mistakes happening to your DNA all the time. But our cells usually pick up on that, and it either changes it or it kills the cell, meaning apoptosis, and the cell doesn't even get a chance to live. But sometimes it does. We can also make changes to our DNA by different kinds of environmental factors. And so when environmental factors are changing our cells, this usually happens during the growth phase. And so when the cell is growing right after it has done mitosis. And so if changes happen during the growth phase, notice that if DNA changes here, now it's going to go into S phase and it's going to copy that bad DNA and it's going to pass that bad DNA on and keep on going. And so that can cause mutations, that can cause cancer. And so these changes can happen by UV light, so being out in the sun for too long. If you get a really bad sunburn, that hurts your cells, and that can cause something like skin cancer. Radiation can also affect your cell cycle. So if you're exposed to a lot of radiation, or if you're interested in learning more about this, you can take a look at what happened in Chernobyl over in Russia. It was a large factory, kind of like Hanford, that had a large explosion, and this was quite a while ago, and organisms still in that area are still affected by this, and so the crazy plants and animals that are mutated. This can also happen by chemicals in our environment. So the chemicals that maybe are in the food that you eat, or in the makeup that you wear, or in the chemicals that you use for cleaning around your house. Those can have effects on you as well. So some impacts of mutations. Not all mutations are bad. Some of them can be beneficial, meaning they have a positive effect and is going to help an organism increase their chance of survival. So maybe there is a mouse that is randomly born with a different hair color that helps them blend into their environment better. That means they're going to increase their chance of survival, which means that's a beneficial mutation for them. Another example could be disease resistance. Some people are born with this disease resistance where they don't really get sick. Or there are some people born with a disease resistance where they are immune to the HIV virus. And so those people are being studied to figure out how can we beat HIV. Here's an example of plants. This plant has been mutated to have a beneficial mutation of disease resistance, which means the crops can last longer and be healthier. Whereas here's the normal plant that can be affected by that disease. Some mutations can be harmful, meaning it's a negative effect, and it could cause the organism to have a lower chance of survival. These are things like genetic disorders and cancer. One genetic disorder is called sickle cell anemia. And sickle cell anemia is a disorder where your red blood cells no longer look like nice fatty red blood cells. Instead, they're this C shape, this sickle shape. And so people who have sickle cell anemia have a higher chance of blood disorders and heart disorders because these sickle cells can get kind of caught up together and cause blockages in their blood arteries and veins. And so what happens with sickle cell anemia is that here's their normal DNA sequence, and it gets mutated, it gets changed. And so notice that instead of GAG, now it says GTG, which means there is a substitution that occurred, which means that instead of producing the normal protein of GLU, now the sequence is different. And now it's going to create a different protein, VAL, which means its whole protein sequence is changed. It's messed up. And now they're going to produce blood cells that are not the right shape. Sometimes mutations can do absolutely nothing. They are neutral mutations. It may cause a change in the organism, but it doesn't really increase the chance of survival or decrease the chance of survival. An example of that is blue eye color. Some people have blue eyes, and blue eyes is actually a mutation. It doesn't do anything for the organism, it just changes their color. Sometimes these natural mutations, or these neutral mutations, will make a change in the DNA sequence, but it's still going to make the same protein. Remember on our codon table, we have some amino acids that have 5 different codes for them because now it's a built-in mechanism for your body where if you have a mutation, it can still produce the same amino acid and it can still produce the same protein. So here's our final chart. This is what you will record in your notebook. Sometimes we can make man-made changes and mutations. So for example, one change that we have done is we took this fish, this is a naturally fluorescent fish, and we took a gene from the jellyfish that makes the jellyfish glow, and we inserted it into the fluorescent fish, and we created glowing fish. So they're a nice novelty, they're fun and everything, but they were actually created as a way to detect pollution in water in areas where they couldn't afford water tests to see if their water was okay to drink. And so these fish were put in the water, and if they started glowing, it meant that there was some kind of pollution in the water and the water was bad. But now you can just buy these glowing fish and have fun with them. So your assignment, along with this video, is you're going to be given a reading called Unit 3 Genetic Mutations Introduction Worksheet. With that reading, there will be 2 pages. One page is the reading, front and back, and the second page has an anticipation guide and then questions on the back of that. You are going to start by answering those anticipation guide questions without reading the article. See what you remember from learning about mutations just now or something in the past. Then you're going to silently read through the article and highlight words that you do not know and make a list of those words on the page that will be written on the board for you. Then you're going to read the article again and make annotations. You need to make at least 3 annotations, meaning you are going to include anything that you highlight, you need to make a comment about. So maybe there are words or phrases you don't know, or you have a question about, or something you find interesting. Write a comment for it. Then you are going to go through the anticipation guide again, re-answer those questions, and make any questions that are false into a true statement. And then you can answer those questions on the back of that anticipation guide. Remember that the more things we do with a reading or some kind of material, the better you're going to learn it and the better you're going to remember it. So please don't just skip through and answer the questions. Follow through this process because the more times you read it, the more times you're exposed to it and using it, and the better you'll be able to remember it.